All right, fabulous. Can I do a audio check? I'm using headphones here to see how's this sound, Tom?
It's very clear. Thank you.
Awesome, awesome. All right, well, thank you, Tom. Excited to present a trial here. Let me get oriented. Let me share my screen. Um,
While Scott's getting that, I just want to remind folks that we have a Q&A function. So if you've got a question, you can use that feature to type it in, and we'll either answer it live or, if it's more appropriate, we'll send you a direct response. And so just make use of that. And of course, the chat feature as well, if you just have something more general you'd like to share.
Yeah, so
yep,
we'll keep an eye on that and interrupt
yeah.
Scott if necessary.
Yeah, please do. Please do. So this will be a mix of facts, capabilities of facts, simulation, an example trial, what sort of happened. The trial's over, so you can see the end of the story to this, and then the trial and facts. So let me get to the story here. So, in October of 2012, Lilly announced positive results of phase 3 trials of dulaglutide in type 2 diabetes. Let me get rid of that. So they announced positive results of what they called their award trials for dulaglutide. Positive results in award 1, award 3, and award 5. Meeting the primary endpoints, superiority for HbA1c lowering for both doses, 0.75 and 1.5 milligrams. That's going to be part of the story here. Demonstrates statistically superior reduction in HbA1c at 52 weeks compared to sitagliptin in that, and award 1 and award 3. It talks about the 5 trials that we're running, and it makes reference to 1.5 milligrams here in all of those. I'm going to focus on award 5 at the bottom, the award 5 trial. And you can see the 1,100 patients total in the award 5 trial. That's the trial we're going to simulate in facts. It has an interesting aspect of award 1 through 4 is that the generation of this 1.5 milligram dose in award 5 is what spawned the other award trials. So that selection of 1.5 milligrams to be investigated in these other aspects of the trial, and additionally in a cardiovascular risk trial that ended up showing cardiovascular benefit, came from award 5. So this was an announcement in October 2012. Further the story in 2014, you can see this report that FDA hands Eli Lilly a big win, okays dulaglutide for diabetes. So it's approved for the treatment of diabetes at this point in 2014. Interesting part of the story is that they project in this 1.3 billion by 2020 for sales. Originally, they projected
Scott,
700 million.
Could you make the slides bigger, Scott? Yeah, perhaps make it full screen.
How's that?
Good. It's better. Thanks.
Okay. Okay. Good. Excellent. Yep. Okay. So they projected 1.3 billion in 2020. And interesting, so this shows in blue the 2020 sales. It actually did significantly better than that, 4.7 billion. This is Trulicity. So dulaglutide's name is Trulicity, and forecasting 6.6 billion in 2024. And of course, this is a GLP-1 and the whole story of GLP-1 and weight loss and what's going on in the industry. This is not surprising. So let me, there is, if you want to read more about award 5, here are the statistical details. talks about the design, the simulation, so you can go read this article. There's also a companion article to this, which talks about the operational aspects of this trial, the Award 5 trial. Let me get out of that. Oh, actually, no, let me go back in there. Okay. So very quickly, I'm going to give you the PowerPoint aspect of the design, and then we're going to jump into facts in this. The Award 5 trial was a seamless phase 2/3 trial. The first part of the trial was a burn-in trial of 45 patients. Actually, it was 7 doses of dulaglutide. You can see those in red. An active comparator, sitagliptin, and placebo. There was an initial burn-in where these initial 45 patients were enrolled. At that point, an interim analysis was done in the trial. The only role of that interim analysis was to set randomization probabilities for the 7 doses. Placebo and active were fixed during this phase 2 part of the seamless phase 2/3 trial. So it used response adaptive randomization, and interim analyses were done every 2 weeks during this phase of the trial, up to a maximum sample size of 400. Again, placebo and sitagliptin were fixed. Interims every 2 weeks. Response adaptive randomization was based on 4 endpoints: HbA1c, weight loss, which of course becomes a really important story within the GLP-1s, blood pressure and heart rate. The huge risk at this point, the huge concern is that were there cardiovascular issues with these drugs? And would the blood pressure and heart rate increase lead to failure in the eventual cardiovascular risk trial? Fen-phen and other types of weight loss, other types of diabetes trials had cardiovascular issues. And so this was the concern. And so they wanted to make sure they got the dose just right. These were all combined together into a utility function in order to select the best dose of the 7. Once 200 patients were enrolled, the trial could make other types of decisions other than RAR. At any one of these 2-week analyses, and these 2-week analyses were done every other Wednesday, every fortnight, every Wednesday, and it could decide to go to phase 3, and it could select 1 or 2 doses. If that was done, it would spawn other phase 3 trials, and you saw that happened, and it spawned the other award trials with the dose selected in this phase 2 part. It could stop for futility, or it could go all the way to 400, and then the rules were changed slightly at 400 to go, but that was the last point at which it could seamlessly go to phase 3. It could also stop enrolling, and this would be a phase 2 trial, like a traditional separate phase 2 trial. If it goes to phase 3, it's fixed sample size. It's actually the sample size was driven by phase 2, powering from phase 2, but that randomization would be fixed. So the phase 3 part looks like a traditional type of trial. There were no interim analyses during phase 3, but the sample size that was selected and whether it was 1 dose or 2 dose, possibly selected, was adaptive based on phase 2. The final analysis of the trial was an ANCOVA model on HbA1c with a one-sided alpha for the selected target dose with a 0.020, one-sided alpha. That was demonstrated by simulation to control type 1 error for the selection of the dose. It used all data from the particular arm selected in the end of phase 3, and it used this 0.020 ANCOVA. And this was approved by both EMA and FDA. Okay, I'll show you in facts the utility function, but there's a utility function for combining together these 4 endpoints. So the algorithm looked at these 4. It addresses predictive probability of statistical success. And that was a non-inferiority analysis to sitagliptin at 12 months for HbA1c change for that. As you saw, the final analysis of that ended up, oh, sorry about that. Spam is calling me. The final analysis for that ended up demonstrating superiority. Okay, so I'm going to show you in facts almost exactly the trial. There's a few weird things that went on in the trial. Facts did not exist when this trial was created. So it was not simulated in facts. Custom code, as Tom referred to, that was created for that. Interestingly, a good bit of what's in facts comes from this trial. We took a lot of the aspects of this Award 5 trial, and it became the skeleton of facts. So not surprisingly, it can largely simulate that aspect of it. Within that, some of the details have never been disclosed. If you read the Award 5, it doesn't actually tell you a few aspects of this. So it's not exactly the same trial here, but in large points, you'll get the general idea of this. It took 9 months to create this trial. That was the iteration of the design. It started as 3 doses. It iterated to 7 doses with response adaptive randomization. The original phase 2 separate trial that was going to be run would have been 3 doses, running out to 12 months, look at the data, make a decision to do phase 3. If it's positive, run phase 3. We iterated for 9 months. We went to regulators, writing custom code to that. You can now simulate this in a couple days. So you could go through this process of iteration and design building with facts rather than custom code much, much faster. That's sort of the role of facts. Okay, so let me jump to facts here. And what I'm going to do is I'm going to save this as a new file name, temp, so I can kind of mess with it and not lose my simulations within this. And I'm going to walk through this. I'm going to start with the phase 2 part of the trial. So I described largely the structure of the trial. I'm going to start and build phase 2. Then I'm going to jump in and use the staged design within facts to simulate both stage 2 and stage 3. Stage 1 and stage 2, phase 2 and phase 3. I'm not going to walk through and type in all of the stuff in facts. It's not exciting for anybody to do. Even with as fast as facts is, this is reasonably, it takes a bit to run these simulations. So I have 100 simulations pre-simulated to look at in this. Okay. So the study tab, I'm doing just the first part of this. I'm enabling adaptive features. The maximum size of this can be 400 enrolled in the trial. I've inputted my 7 doses of dulaglutide. I'm sorry, wrong tab. I'll do my treatment arms. I've put in, and this is Lilly. This was originally had its LY some number to it. 0.25, 0.5, 0.75, 1, 1.5, 2, and 3. Sitagliptin as an active comparator. It has placebo as a control. I'm going to mess with these a little bit in facts, and I'll sort of explain why and the role of the trial. Overall maximum sample size of 400. The visits where we get data overall are 2 weeks, 4 weeks, 8 weeks, 12, 26, 39, and 52. The different endpoints have different final time points in that. So this is a master list of possible visits. But not all the endpoints are collected at all of these, and I'll sort of explain that when I walk through the endpoints. So the treatment arms, the 9 treatment arms, 7 of them active, the visit schedules in here. So I'm in the multiple endpoint core design engine in this. So the multiple endpoint allows you to simulate, as it describes, multiple endpoints in the trial. You can use as many as 4. And I think that probably comes from this trial. It's the most I think I've ever used in an adaptive trial where explicitly the adaptations are driven by multiple endpoints. It's not uncommon. We do an efficacy measure and a safety measure. You might be doing a Project Optimus trial where you're looking at tolerability and ORR. You might be, so it's a number of things. This one used 4. We've added these 4 endpoints here within this. Let me go to this. So I get to add the endpoints in here, and I've added them. I've named them. You can go in here and name these different endpoints something. HbA1c, weight, heart rate, and blood pressure. So HbA1c, I'm going to show you the utility function in a second. HbA1c, it's a continuous endpoint. Decrease is good within this. I am going to use longitudinal modeling, and from the master list, I get to click which of those visits I collect HbA1c at. And I'm collecting it at 2, 4, each one of the visits I collected. The last of those is going to be the primary time point that I'm interested for that endpoint. You'll see for weight loss, that's going to be 26 weeks. So the interesting thing about this is HbA1c is the only one of these endpoints that I'm comparing to the active comparator sitagliptin. So HbA1c's non-inferiority to the active comparator. All these others are compared to placebo. And placebo only goes for 6 months. At that point, patients are put on to sitagliptin, the active comparator. So the exposure to placebo is only 26 weeks, but you're allowed to do that in facts to specify that I get to see those endpoints only up through that. When I do this simulation, the only role of the active comparator is HbA1c. It just makes it easier within facts to tell facts I'm comparing to the for the utility function to the placebo, and I simulate the placebo's HbA1c like sitagliptin. So it's just a straightforward way to do that in facts, even though it's the sitagliptin arm that's there. So this trial, through its iteration, as we designed this, originally we started to put a whole bunch of rules in there about how do we pick the dose from phase 2 to phase 3. We ended up with a whole bunch of rules. If HbA1c is here, we want to do this, but what about heart rate and blood pressure? And originally there's resistance to create a clinical utility index. But this became really the only way to do this. So this mirrors almost exactly the clinical utility index that was used in the trial. And you can see up here, this is going to multiply the individual pieces that come from each of the 4 endpoints together. And I'll show you how that comes about. What we're going to do is create a utility for HbA1c. So if a dose could have this value relative to sitagliptin, then it gets that utility, and it's multiplied by the utility for the other endpoints. It can go up or down, and it can be zeroed out if side effects are bad. Safety, heart rate, and blood pressure are bad. The value of the dose can be 0, which is the minimum of this. So HbA1c, a value of 1, that's been a normalized value, and this is a range of non-inferiority from 0 to minus point, in this setting, 0 is here to even an increase of 0.25 because it can show non-inferiority. So a dose that's non-inferior in that range, equal or non-inferior to sitagliptin, has a value of 1. That increases if it starts to be superior at a pretty high rate, because showing superiority is that much more value. Still more value if you go all the way back to a -1, and then decreases beyond that were thought not really to have additional value. So or value of 4 is the highest this utility function can be. And that's specified in here, and it graphs it and shows you that graph. So this is really the comparison of doses within that. Now, weight is kind of a marketing endpoint. It didn't need to show anything on weight to be FDA approved, but the thought was, if it increases weight, it's hardly going to be used. It has little value. If it decreases weight, it has more and more value. So this function gets multiplied by the value for HbA1c. So you could as much as increase its value by 1.5 times if you had weight loss bigger than -5 or better than -5 at 26 weeks. And you can see this increases up to a value of 1 right here at 0. So if there's no difference in weight, you get the multiply by 1. As you start to get weight loss, it increases the value of the dose. If it increases in weight, this goes down, and above 1, it has 0 value because it's not going to be used. Heart rate and blood pressure are very similar to weight. You get full value. You multiply by 1. If the heart rate increases by 3 or less, that was considered minor. If the heart rate increases by more than 3 beats per minute, up to 6, you get linearly decreasing value down to 0 value. If it's greater than 6, it was thought that they wouldn't even run the cardiovascular risk trial, that the concern was it would increase cardiovascular risk. And the same for blood pressure. It looks very similar to heart rate in its functional form. So that creates the clinical utility of a dose. And the major role of that are comparing 2 doses. Which 2 doses would we rather have? To back off, in the creation of this, we showed the design team multiple profiles and said, suppose you had a dose that did A, B, C, and D on these 4 endpoints versus one that did X, Y, Z, and Q. Which would you pick? And then we showed what the clinical utility said around them. And the first time we created one of these, it worked pretty well, but there were scenarios where they said, no, we'd rather pick this one. So we altered the utility functions in a way that it matched what they wanted to pick. And then we continued to show them more and more, where largely every time we showed them a profile compared to another profile, they picked the same thing the utility function picked, even to the point where sometimes they didn't pick it. We showed what the utility function said, and they said, I think that's right. So they became completely agreeing with the utility function. It took a while to iterate that to get the team to agree to that. It became locked in the trial that this was the way the dose was going to be selected, and it could not be changed. The DSMB, I'm not going to say much about the monitoring of this, but the DSMB monitored this for safety, but they couldn't change the decision that was made. The utility function was making the decision around dose. Okay.
So Scott, was there any need to get buy-in from the regulators on the utility, or was it just a internal decision making?
Yeah, it mostly was internal, but the heart rate and blood pressure was actually helped by regulators. When we went to them, we actually had only one of these in there. And in the discussions with them about the appropriate values, largely the 6 and 2.5 came from regulators. So there was an interaction with regulators on this, yes. Weight and HbA1c, no.
Thanks.
So let's jump into, I'll skip virtual subject response, execution. I'll go to the design. So this is the phase 2 part of the design. We have 4 endpoints. We created dose response models, and these are modeling the 26-week time for 3 of these and the 52-week time for the other one. Turns out the longitudinal model here is critically important. But so in this section for each of these endpoints, we pick the dose response model and the longitudinal model for this. HbA1c, the models, by the way, were very, very slightly different than this, but these are the best representations of this. Is a hierarchical model with a mean function for the hierarchical model that was a logistic. So what this is each of the 7 doses has a logistic curve as the mean expected to decrease, though we don't know its effect. And then each of the doses around this is hierarchically set on that mean function. The closer it fits that curve, the more they shrink towards the curve. And we set up the parameters to that around the expectation of these doses, the expectation around the slope with a mean and standard deviation. The controls and sitagliptin are set separately. Here's where this weird part that the control, the placebo, is not expected to have a -1 HbA1c change. But I'm doing that because the utility function in facts is against the control. So I simulate the control with a mean of -1 within that setting. That becomes really the active comparator plus the placebo itself, and it works perfectly in facts. Weight, I'll come back to the longitudinal models. Weight has a similar, the expectation of this is that it's going to be monotonic, but it's hierarchically around there. It doesn't force monotonicity around the doses. So it can fit non-monotonic curves. Within this, it's quite flexible. The heart rate within this one, I didn't mess too much with these because these become reasonably well defined within this setting. These were normal dynamic linear models within them for heart rate and blood pressure both. They're reasonably noisy endpoints, but we get multiple measures of them. And I guess I'll jump in. Longitudinally, we assume that it's measuring on drug heart rate and blood pressure. So it's the average over those 26 weeks. So in the longitudinal model, FACS knows it's getting these multiple measures. All we do is we force each one of these to have the same mean, and it estimates that mean over the 26 weeks. So it's an average blood pressure and it's an average heart rate for the longitudinal model. So each subject gets multiple measurements of that, and it's estimating what is that average over the 26 weeks. That's the estimand. And you can have FACS do that by setting the longitudinal model to be 0 with essentially no variation. Really important was the HbA1c longitudinal model, which we selected as a time course hierarchical model. What that is it assumes a hierarchical distribution for each patient as to what HbA1c reduction they're going to see after 52 weeks. And then it estimates the fractional change over the visits. So each one of these visits, and this is e to the alpha, creates a fractional aspect of the total change. within this. And this is e to this parameter, so e to the minus 0.03 is almost 1 within that. And so you almost get 100% of the change at 39 weeks. 26 weeks, you see you get most of the 52-week change. This was estimated from previous phase 2 data on other compounds in diabetes as to the rate of HbA1c and the relationship over time. We spent a lot of time with historical data, and these priors have a reasonable amount of informativeness in the longitudinal model of the expectation of what happens within a patient for HbA1c. Weight, very similar. We also had historical data where we put informative priors on the longitudinal pattern, also the time course hierarchical, and FACS allows you to pick different sort of models to this. We picked the time course hierarchical model to this for weight loss. Hey, the dog doesn't like this modeling. Okay. Within that. So this is the mathematical structure of the models that are driving the adaptations. Now the interims here. I set it up 4 weeks to simulate. I could set it up here every 2 weeks to simulate. It does the first interim at 45 subjects enrolled. That's the burn-in period for this. Within that, and then every 2 weeks, it's going to do interim analyses. And it sets it up information at the first interim is 45 subjects, and then every 2 weeks during the course of the trial. The maximum for this is 400. What is the allocation? It has fixed, it enrolls 5 for each of the 9 arms. That's 9 times 5. After that time point, it fixes 2 for placebo and 2 for sitagliptin out of every 10. This is during this phase 2 part of the trial. The remaining 6 of the 10 are adaptively allocated to the different doses, the different LY doses. So what happens at the adaptive analysis is that it fits the posterior distribution based on the data it has. You can see nobody's going to be at 52 weeks. Nobody's going to be at 26 weeks at the first interim, and how important the longitudinal model was for forecasting that data. It's going to fit the distribution, and then based on the utility, and 0.6 became a really valuable one. Essentially, Lilly decided that if one of these doses is greater than 0.6 utility, or likely greater than 0.6, they wanted to go forward. So the adaptive allocation based on the utility function that combines these 4 endpoints together. That creates the relative randomization over the LY doses for that. If individually those probabilities were less than 0.05, it would be zeroed out for that 2-week period, but it could come back 2 weeks later. So it wasn't dropped within that. It was zeroed out for 2 weeks and could come back. All right. Then the decisions. No decisions. We're doing a lot of interims. We start at 50. We do a lot of interims up to 200. At 200, we can start making decisions. So there's no futility. There's no graduation to phase 2, no success in this phase 2 trial. But you saw in the original part, if this is successful, it's going to move seamlessly. And on Thursday, it's going to be a phase 3 trial. Wasn't quite 1 day, but it was a very short period of time upon hitting graduation. It would now be phase 3, and it was fixed randomization for the selected dose or doses and the control arms. What were those criteria for futility? If the probability of winning the phase 3 part of the trial. So we created a sample size for phase 3. And if the probability it would win that for the maximum utility dose was less than 5%, we would stop for futility. Or if the posterior probability of having a utility of at least 0.6 was less than 5%, the trial would stop for futility. And that was either or of those. All of the patients would be followed, they'd read out, but the trial would stop enrolling and it wouldn't move to phase 2, to the phase 3 part. If these criteria were met, both of them were met, and we had at least 200 patients, it would immediately go to phase 3. And it would pick, I'll describe how it picks the dose as I move this to the seamless 2-3 trial. So if the greatest utility dose, so what dose the algorithm thinks is the maximum utility dose, that was the role of the utility function, is greater than predictive probability of winning phase 3 for that dose is greater than 85%. And that's a non-inferiority analysis compared to sitagliptin. And the probability of being above 0.6 is greater than 0.8, it was going to move to phase 3. And that was the algorithm. At the final 400 patients, so when 400 patients are enrolled, a final analysis in facts is done. If the probability of winning phase 3 non-inferior is above 0.6, so it lowered the threshold at 400, it would graduate to phase 3. So that it had this lower criteria for going, but it would spend more time in phase 2, learning the right dose, refining it, getting longer exposure. But if it was above a 60% chance of phase 3 success, it would go to phase 3.
Scott, you have a question in the QA.
Okay.
The Q&A. Asking, when you simulated the operating characteristics across the 2 phases, did you treat the futility analysis, the early futility, as binding or non-binding in calculating, and did you worry about that distinction?
We did, yes and no. So in simulating the trial, and that was an important reason why it was locked within that. It was treated, the decision to go from 2 to 3, especially this, for example, and the non-decision was treated as binding in the simulation of this trial. We did simulate it where we turned that off and it automatically graduated. But in the 0.020 nominal alpha, this was treated as binding. When we went to the DSMB, we told them that, that this is binding. And that's an important aspect of the algorithm. This is rarely done, but yes, in this trial, it was binding. Okay. I am going to, I don't want to save that, no, but I want to open, sorry, I'm going to come back to this. I'll very quickly open the phase 2 trial that I originally had open. Recent project, phase 2 facts. Okay, this is exactly the same trial, but I have simulations. Oh, so in simulating this, it gets reasonably complex. I've created 5 scenarios of HbA1c, 3 of weight, 2 of heart rate, blood pressure each. Within that, if you multiply, that's a lot of scenarios. So you get to create composite scenarios to these. So I only wanted to deal with, in this demonstration, I only wanted to deal with 7 different composite combinations of those as interesting ones. By the way, when we submitted the simulation report to regulators, we had 300 null hypotheses that we were simulating. within the scenarios. Just null hypotheses within that. So HbA1c, bad means that it has no effect whatsoever. Sitagliptin has -1 as the estimate, and this is 0. So it's awful. The null hypothesis is that it's 0.25 worse than sitagliptin. The non-inferiority margin was 0.25. So every one of the doses, and this is kind of the hardest null hypothesis, is within 0.25. Within that estimated 0.8 standard deviation. Equal means all of the doses are equal. Increases where it's inferior to sitagliptin at the low dose, increases up, becomes equal at 1, a little superior at 1.5, and you can see is superior at high doses. That's HbA1c. And then there's a great scenario where it increases even up to -2. So it's quite a bit better than sitagliptin. But then how is that compared to heart rate and blood pressure? So here's a scenario where there's weight gain, which is bad. There's no change in weight and weight loss. So you can see up to -5, and that was the point where you're getting 50% more value by that for high doses within that. 4 is the standard deviation. And then heart rate and blood pressure. There's no effect on heart rate, which is great. Or high, meaning even at high values, the utility of that's going to go to 0. And that value would have no value at high doses. So you can see as those get compared and composite together, we can create interesting scenarios. So I'll go to the simulations really to show some of these scenarios. So here's a scenario that is, I'll look at 100 simulated scenarios. Here's a scenario where the black here shows the true utility of this, where it combines together scenarios that I've named good. That's good HbA1c, but it has heart rate issues at high doses. So these values do not have high enough or very high HbA1c changes in that. The truth is these first 3 doses have low utility, not 0.6, by the way. Then it increases up, and these doses 1.5 and 2 have high utility, very high utility. Above 0.6 is considered, let's go to phase 3. And then LY3 has no value because the heart rate increases. And we can see that a little bit with the different endpoints. So this is the HbA1c effects. So it's monotonically getting better. It shows over the 100 simulations what the estimate of that curve is, and then the estimate for sitagliptin in control. The second endpoint, you can see weight loss. You can see heart rate. Now we start to have issues, and it gets to values that are too high in the simulation, where blood pressure has no effect within that. So the combined of that is this. You can see the average sample size. So you can see response adaptive randomization trying to hone in within this region. It's decreased here. It's decreased here because of efficacy. And it hones in this middle region of the response adaptive randomization part. Subject allocation. Overall, the let me kill this. It's in my way. Okay. 68% of these hit early success. 12% would hit success at 400. So this trial was exactly 80% powered for this good HR scenario in phase 2 to graduate to phase 3. If the effect was the null in my 100 scenarios, all of them stop for early futility. So in that case, you can see the overall utility is 0 because it's not non-inferior. to that. You can see the subject allocation. It's looking for something within that scenario, within that, but every one of those trials stopped and did not graduate. If it was solid, so this is where it's exactly equal to sitagliptin within that scenario. It does have, I think, it does have weight improvements. It did go to higher doses. There's no side effect issues, so it goes to higher doses.
Scott, there's a question which is why in all the scenarios did you use the mean on control the same as the mean on sitagliptin?
Yeah. So that's a facts thing. And it's because when I go to the utility function for this, the utility function, I want to parameterize this relative to control. So 0 is control. And the heart rate and blood pressure, we didn't want to do that relative to the active comparator, but relative to placebo. It was why placebo was in the trial, and the change in weight relative to placebo. So in the utility function, I can't do relative to active comparators that. So the utility function is relative to control in each of those. Now it's fine. All I do is simulate the controls HbA1c to be sitagliptin's HbA1c. So there's no harm in that. And then I have, so the active comparator doesn't actually do anything in this trial in there, but it's there to take up its randomization and aspect. So it's purely the aspect of the utility function being relative to control. The controls HbA1c didn't really have an impact, but the sitagliptin. Sorry, Tom.
Sorry. I was.
Yep. So interestingly,
you're completing your answer, but
yeah.
So we got a second question, which is the rather frequentest one, which was the family-wise type 1 error evaluated with repeated Bayesian interim analysis at alpha 0.025?
So the primary analysis at the end of the trial was, I'll show you this, for example. Here's a scenario, an awesome scenario. I called that awesome within this scenario. So the truth of this scenario is that the 2 high doses have this effect of 6. And that's really HbA1c continues to go down. Weight loss continues to go down. They have weight loss of -5, and then heart rate and blood pressure have no effect. So you want to pick the high dose. There's no negative to the high dose here. This shows the selection of the dose of utility. This shows which dose is selected. So in this, 60% of the time it picks LY3, about 30% LY2, and occasionally these lower doses. That dose that's selected that goes to phase 3 becomes the primary analysis in the trial that compared to sitagliptin. That's tested at 0.020. That 0.020 was demonstrated to control type 1 error at 0.025, family-wise 0.025. That the trial can pick any of these doses, it can cherry pick good data, go to the end, that 0.020 controls type 1 error. Then, of course, there was then test superiority, test other endpoints, but the primary analysis tested the target dose that was selected. I'll show now what I did is I imported this into the staged design. So I took exactly that design you looked at, and that's stage 1. And those are now the graduation criteria to stage 2. So I actually imported it in. You can go into facts and say, import that core design as my stage 1 design. And that becomes graduation now. And then stage 2 becomes fixed sample size. And I set this up. There's actually, you can select a lower dose to go additionally, and the actual trial did select a lower dose to go, but I'm simulating this just a higher dose for summary here. And it looks at, it enrolled 1 to 2 to 2 and to 2 for the other dose. So these were fixed in that stage 2. The final analysis in stage 2, I did something funky here, which FACS allows you to do. This is the 0.020 nominal alpha for the dose that's the maximum utility dose. And that's the one that demonstrated control of type 1 error. I set that up as futility. So if that's not met, FACS calls that a futile trial. If that is met, then it looks at success, and I called superiority, and I use the posterior probability of this. I called this, I use this as a measure of superiority within the setting, and above 0.975. So what that means is FACS could have this trial end up between futility and success. FACS calls that inconclusive, but I know that that means it met non-inferiority, but not superiority in the trial. Okay, so now when I simulate this, I'll go to this awesome scenario where we know that it always graduates, and I can view stage 2 graphs of this. You see the sample size is now, the average sample size in this scenario is, you can see this is up near 1,000. You can see the control on average enrolls 300-ish within that scenario. You can see the doses selected, and we looked at phase 2, and that's exactly the doses selected. We can then look at the outcome of those. And every one of these met superiority within the trial. And you can see by dose selected, it met superiority in that. None of them were even inconclusive, which meant non-inferiority. If we go to a less good scenario here, and we look at this scenario, and this is the scenario where it could actually select an unsafe dose potentially here. Largely, it picks the right doses to go to phase 3. It can make a mistake in this setting. You can see the outcome by arms selected. You can see sometimes it's inconclusive. Sometimes it never goes. So about 81% of trials go to phase 3, and you can see the fraction that were met. This means it met non-inferiority. This means it also met superiority. And you can see that by doses here. LY3, non-inferiority. If it picks LY 1.5, it met superiority in each one of those. And so you can see outcome by the selection of those within the scenario. If it's unsafe, if it's unsafe, it never goes to stage 2 in that scenario. And you can see heart rate and blood pressure both becoming an issue here. And it has a narrow 1 and 1.5. You can see how often it picks those doses, stays away from high, stays away from low within those. Ooh, I wanted to show this. And you can see the outcome by arm selected. In that large fraction of these being non-inferiority, but not superiority. Sometimes it goes and it loses phase 3. If it picks LY 0.25, it tends to lose because the truth is it wasn't very good. And so you can see that. What happened in the actual trial is that as soon as the sample size was 200 at a Wednesday analysis, it picked 1.5 milligrams. And you saw in in the Award 5 program, the algorithm picked 1.5 within that setting. And that spawned these other trials. It also took 0.75 milligrams. It could take a dose more than 2 doses away from the target less. If that dose looked good enough, and 0.75 also looked good enough, by the way, 0.75 also demonstrates superiority to sitagliptin. So it selected 2 doses and it moved to phase 3. It spawned the other trials within this scenario. And this became Trulicity's marketed dose. Another interesting thing, which has been publicly disclosed, is that during the course of this trial, the DSMB voted to stop LY3 because of heart rate and blood pressure. The interesting thing was the algorithm had zeroed it out for multiple interims. So the algorithm had already diagnosed the issue and it was giving 0 randomization, but they officially dropped it from phase 2 that 3 couldn't be used anymore, but it was already implemented in the algorithm and it was set up in that and selected 1.5. By the way, I think since they've gotten 3 approved for some scenarios within diabetes. I don't know enough and don't take that, but I think it's actually is used in some cases. Okay, sorry, it was a whirlwind hour of facts. I really wanted to demonstrate the multiple uses of multiple endpoints, the utility function within this, the seamless 2-3 trial within there. I didn't focus a ton on let's simulate the null hypothesis, looking at type 1 error controlled within that. It was a huge part of the interaction with regulators, but is less interesting in sort of the facts aspect of this. Any other questions on there, Tom or Nick?
So we've got one more question in the Q&A. I'm going to just preempt it with a simple question. Do you think this is actually one of the most complex trials ever designed, and also at the same time one of the most successful, not just in how well the drug did, but actually in how well the design performed?
It's certainly one of the most complex I've been involved in. And part of that is the multiple endpoint aspect of it, the clinical utility aspect of it, the frequency of the interims being every 2 weeks during the scenario of this. By the way, each of these interims ran every 2 weeks. Tusela ran this trial. Tom was at Tusela at the time when this ran. And it never needed to be adjusted. So the algorithm ran completely unadjusted. DSMB zeroed out that dose, but that didn't affect the algorithm. The algorithm picked the doses. They were not changed. It moved to phase 3. So in terms of that, I think it was incredibly successful. And it's hard to sort of not think that this was successful. The other part of this that Lilly has said, this wasn't me or us, that this sped up timeline 6 to 18 months, which means end of life, patent life. With that, at $6 billion a year, that's kind of a big deal to them within the settings. The other part of it is the seamless 2-3 trial, the algorithm, all of that sped it up. But interestingly, I don't know if 1.5 milligrams would have been in their phase 3 trial had they run a traditional 3-dose phase 3 trial. Maybe they pick, 0.5, 1, and 2. Maybe they pick, and 3 is part of that. I don't know the 3 doses that would have actually been picked. So the value of this within terms of most successful, I think there's a lot of measures of this that were very, very successful, but one I don't even know is would 1.5 even exist as a dose without the ability to do 7 doses, response adaptive randomization. If they don't do RAR, they would have done 3, maybe 4 doses. But because of RAR and phase 2's honing in on the right doses, they did 7.
Thank you. I know we've overrun a little bit, but can I just ask you the one other last question that's on the Q&A? The questioner asked about family-wise error rate earlier. It says their experiences that calculating the family-wise error rate under binding and non-binding futility makes very little difference to the value. That's the value of the alpha test at the end when you've only got one interim analysis. But the impact on the value of the family-wise error rate may be more pronounced if the interim analysis outcome is different from that of the primary outcome, as it is in this trial. What's your take on this, and do you have any advice?
Yeah, I think in this trial, it was different in part because it was a very aggressive futility rule within that. I think traditionally you do one interim. It's an O'Brien-Fleming stopping boundary. It's typically very, very conservative. Largely, you get rid of it. It doesn't make much difference. What was interesting here is futility was driven not just by HbA1c. And in many ways, it was driven by side effects and weight loss, things that did not affect the primary endpoint. The other part of this, one of the biggest parts of this, is that it didn't cherry pick the best HbA1c. Even the actual trial picked a dose that wasn't the best HbA1c, but it optimized the 4 endpoints. So it was an interesting aspect that the selection itself was not picking the maximum of the 7 doses, depending on the other endpoints, which also affected how much cherry picking impacted the type 1 error. The other part of this is regulators did say, we'd be more comfortable as long as the phase 3 part that's fixed makes up at least 70% of the sample size. So the adaptive algorithm knew that the phase 3 part had to make up at least 70% of it. They said they would just feel more comfortable under that setting. So it was simulation control. It had multiple interims, the weird selection based on a utility, but also that phase 3 had to be a substantial part of the overall sample size. This was also one of my favorite regulator quotes is Rob Hemmings, when presented with this, said, "I've run out of reasons to say no." Which I thought was just awesome.
And have we done any trials using utility functions since this?
Yes, though I think largely they are 2 endpoints, an efficacy endpoint and a safety endpoint. But we have done. I haven't done 4 since this time point. Project Optimus is getting more interested in this, of course, as finding the optimal therapeutic dose as a combination of these. I can't say as though I can tell you exactly how to do this, but that's becoming more and more interesting to use a utility function.
Well, that's a great talk, Scott. Thank you very much.
Sure. Thank you all.
Folks, and we'll be sending out the link to the recording so people can share it more widely. So I hope to see you next week.